Okay, so next we need to talk about how amino acids are chiral. So if we think back to carbohydrates, we had talked about glyceraldehyde or Fisher projections with our sugars and how those were chiral. So we had the two enantiomers of those kinds of sugars. We can also draw Fisher projections for amino acids. So for example, let's switch back to blue. For example, we can draw our amino or a carboxylate at the top. There we go. Draw a line down. We have one chiral center because we have one carbon that has 4 different groups around it. And then we'll have our nitrogen, our hydrogen on one side, and then we have our R group down on the bottom. So if we have our Fisher projection of this amino acid, we can also have our enantiomers. So let's draw a dotted line, or you can draw that mirror plane. Let's draw the enantiomer. So again, we're just drawing the mirror image. So we can have both different enantiomers of amino acids. We name these a little bit different. We had L and D. Actually, it's the same. L and D for our sugars. We also have L and D for our amino acids. So this group on the bottommost chiral carbon from the carbonyl is on the L side or on the left side. So it is an L amino acid. AA is amino acid for short. And then over here we have the right side. So we have a D amino acid. In general, only the L amino acids are incorporated into protein. So naturally, only L amino acids are in our bodies, really anywhere in nature. There are no D amino acids in nature. However, synthetically produced amino acids can be the D enantiomer. Also, one thing to note with the chirality of amino acids is that there is one amino acid that is not chiral. So if you look back through your notes, the one that is not is glycine. And if you draw that Fisher projection of glycine, you'll see why. You can have your amine and carboxylic acid, and then your hydrogen on one side, and then your R group is also a hydrogen. So you have 2 groups that are both the same. Therefore, this is not a chiral molecule.